Hello, everyone. Uh, Good evening and welcome to today's session. So, we're gonna wait for 5 minutes so that more people can join in, and then we're gonna start with our today's lecture. I guess we can start our discussion for today. So, if you remember, last time we actually discussed about the fluid mosaic model, right? And what were the basic properties or the characteristics of fluid mosaic model, why it is called fluid mosaic model, and how we actually came across the property of fluidity, how it was experimentally determined, as well as mosaic nature of the fluid mosaic model, how it was identified. So we ended our discussion with the experimental evidences or the experiments which were helpful in actually coming up with the fluid mosaic model or the fluid and mosaic property of a biomembrane. So by now I guess it must be very clear to you guys that membrane has major component belongs to lipid and protein, but we cannot say this absolutely, because depending upon the function of a cell, the components of the membrane remains same, but the composition varies, right? In our previous discussion we actually commented on this, that certain membranes, such as membranes from mitochondria or chloroplast, they are, the concentration of protein is more in comparison to other membranes. And why is that so? Because the membranes of these organelles are highly functional, and they are like functionally very diverse also, right? Hence the components, that is lipid, protein, cholesterol, carbs, they remain same, but their composition varies depending upon the function of a cell or the different cell types. Okay, so now as it is pretty clear that membrane has two major or three major components, that is lipid, protein, carbohydrates, and sterols and cholesterol, right? So today we're gonna dedicate our discussion for lipids itself. By now you guys have some idea regarding what kind of lipids are present in membrane, as during our discussion and even in the informative videos which I have shared, a lot of time, most of the time, or say 100% of the time, the term phospholipids was being used for the membrane, instead of just saying that lipid bilayer. And whenever the name for the lipids is being used specifically with respect to the membrane, phospholipids was the name, right? And phospholipids are basically a type of lipid. So we're gonna discuss today what is the basic structure of a phospholipid, how different phospholipids are present in the membrane, how many types of phospholipids are there. Maybe we won't be discussing different types of the lipids, phospholipids today, but we're gonna end our discussion most likely for the basic structure of a phospholipid, and then we can vary the functional group and see what properties they impart to the membrane, and these lipids whatever they are contributing for the functionality of the cell at the end of the day, right? So lipids as membrane, as we have seen that the type of lipids which are present in the biomembrane are called phospholipids. But keeping the name aside, other thing, when we were using the term phospholipid or say lipid for the biomembrane, we were saying these lipids are amphipathic in nature, right? So what amphipathicity means? So amphipathic lipids, as you can see in this structure here, that they have a circular head. It is the representation or one way to represent phospholipid or amphipathic lipid. Okay, lipids won't be like this. We're gonna see how they actually look like in the coming slides. So if we look here, this round structure is called head, which is polar in nature, in meaning that it will be water favorable or water soluble or water loving, hydrophilic. Okay, on the other hand, there is this long zigzag kind of representation. It is called tail, right? It is made up of it's a acyl chain, so it's just carbohydrates, CH bonds. Okay, and it is non-polar. Non-polar means they are not water loving. They don't like to be in water, and they prefer to be either among themselves or away from the water, right? So they are hydrophobic in nature, correct? So one such representation can be seen here. If one amphipathic lipid molecule is present in a solution, so how they will be oriented? They will be oriented, and this orientation is energetically favorable, right? So the polar head will try to be in the water side, while the non-polar tails will try to be away from the water, and hence away from the interface of water and air in this particular example. Okay, so what is written here? It says that it is quite remarkable that membranes are so stable without any covalent bonds holding them together. The subtle forces driving membrane bilayer stability must indeed be strong, if not obvious. These forces are referred to as hydrophobic effect. The hydrophobic effect is attributed to intramolecular hydrogen bonding between water. So here I am introducing a new concept, right? So till now we were saying that biomembranes are what? They are lipid bilayer in nature, multiple lipids. And if we can go back to our membrane representation, you can see here that there are multiple amphipathic lipids are present, right? And most abundant amphipathic lipid in biomembrane is phospholipid. So wherever I am mentioning phospholipid, I am not only saying that they are phospholipid, I am referring to the lipids which are present in the membrane, because they are amphipathic in nature. And predominantly phospholipids are a type of lipid which are predominantly present in the membrane. So if you can see that there are multiple lipid molecules are present, right? Phospholipid molecules are present, and they are closely packed. But can we see any direct linkage between them? Can we see any covalent interaction like CH bond or CO bond, something like this? No, right? So this property that membranes are rigid structures, they are fluidic in nature, and they are very stable under physiological conditions, it is attributed by non-covalent interactions. Not a single covalent bond or interaction is present while the membrane is being formed or when the membrane is present. Okay, so this is a very remarkable property. Because of this, so if you know, if covalent bond is present, for example here, this is directly connected to the head group, correct? So it must be connected via, most likely by covalent bond, because these are molecules, and molecules, when there is a difference in charge and electronegativity, covalent interactions are the most frequent ones which are formed. So in covalent interaction, it provides rigidity to the molecule, which means it reduces the flexibility of the molecule at the end, right? So if any covalent interaction is present in the membrane, it will reduce its flexibility. And we know till now, and we have already proven that membrane is fluidic in nature. Fluid means it has the ability to flow, change shape, change its consistency, right? So if such rigid bonds are present in the membrane, then the fluidity of a membrane will be affected, or in a way we can say the membrane won't be even fluidic in nature, correct? So all of this information, that no covalent interaction is present, then how come such stable macrostructures are available or formed? So they are formed and they are stabilized by non-covalent interactions. Major non-covalent interaction which is a contributor for making a biomembrane is called hydrophobic effect, correct? So the hydrophobic effect, what it states is that, so if we look at this amphipathic molecule here like this, so water is the polar head is facing the water side, and the acyl chain or the hydrocarbon chain is facing towards the non-water side or the non-polar side, correct? So these opposing forces that the acyl chain, because if you can see, this polar head group is smaller in comparison to the hydrocarbon chain, right? So the effect in this molecule, the major effect will be imparted by the polar head or the non-polar tail. If we go just by the quantity of the number of molecules which are imparting these properties, so by number, non-polar tail is longer. A lot of hydrocarbon is present in this chain. Hence, this particular interaction, that is non-polar interaction, will be more in comparison to the interaction provided by the polar head group. Correct? So the interaction which these tails or non-polar tail provide is called hydrophobic effect. Why? Because these are hydrophobic in nature. The tails, they do not want to reside at the water, in the water, and hence they will try to orient themselves energetically favorable condition, and that energetically favorable condition will be when these tails are away from the water, and hence they will orient the molecules in this structure. This interaction energy or this tendency of molecules to stay away from the water and stay together to balance out the interaction is called hydrophobic effect. Okay, so basically we know that we have an amphipathic molecule which are coming together because of the hydrophobic effect, and hence they are making a sheet like structure. Correct? So in these sheet like structure, the major non-covalent energy or interaction is provided by hydrophobic effect. But wherever there are amphipathic molecules and the association is involved, you know, there will be some favorable interactions and there will be some unfavorable interactions. So if we go back to this, our very easy representation, a favorable interaction will be the one, for example, who is supporting the polar head group to stay in water, right, and the acyl chain to stay in air. Correct? So all the interactions which are favoring this orientation of the molecule will be favorable interactions, right? And all the interactions or energies or associated things which will hinder the orientation of molecule in this way will be unfavorable interactions. Okay, so during lipid bilayer formation, if we talk on a molecular scale, single molecular scale, so basically we can say that there are 5 major forces which are acting on a membrane to stabilize it. Out of these 5 interactions, 2 are unfavorable in nature and 3 are favorable in nature. Hence, majority of the interaction or energies are favorable in nature for amphipathic molecule to align themselves according to the hydrophobic effect, or they will be following the hydrophobic energy profile. Okay, so if we talk about these 5 forces, we can see that what component of a lipid is providing which kind of force. So for example, if we talk about hydrophobic effect, we can see this is a hydrocarbon chain, right? The wiggly line which is represented here is actually this CH, a long chain of hydrocarbon. Okay, so excuse me. Excuse me for a while. Yeah, sorry for that. So the first effect, which is called hydrophobic effect, it's an unfavorable interaction, because if acyl chain is facing towards water, because we are in aqueous system, right? So why water is so important for us? So if you know evolution theory and everything, the first life originated in water itself, right? So from there are certain principles which are there in existence to help us understand the evolution. So we are surrounded with water, even in our cell, if you know, even membrane, right? There is one leaflet which is facing the extracellular matrix or the extracellular side, and one side or the interior of a cell is the cytoplasm or cytosol, which is also majorly aqueous in nature. Hence we are beings made up of water, correct? Hence any molecule, whichever macromolecule you're gonna study, you definitely gonna see its interaction or its behavior in water, because the first life originated in water, and we are also beings which have lot of water, right? So the hydrophobic effect, why it is saying it is unfavorable, because if acyl chains or the hydrocarbon chains are present in water, it's not a favorable orientation, right? Remember this orientation of an amphipathic molecule. So what this interaction is saying, that this acyl chain is present in water here. So you know from our brief discussion about hydrophobic effect that these acyl chains won't be happy in water, right? And hence they will try to orient themselves away from water. So if such orientation is present, which is present because we are water beings at the end of the day, this type of interaction will be unfavorable, right? If we talk about caged tails, so caged tails are basically if a lot of water is present, these water molecules will form a protective cage against these hydrophobic tails, and again it will hinder their coalition among themselves, and hence stable structures won't be able to form, which is required for the formation of a cell membrane. Okay, now if we talk about head interaction, so you know, as I have mentioned, that head is a polar molecule, right? So polarity means it has some charge associated with it, or whenever another group comes near to a polar entity, it will try to polarize the other molecule also, right? So opposite attracts, remember this. So head interaction, in which one side of one molecule is partially negative. On the other hand, because of this, it will induce, or maybe say other molecules also partially charged, and they will attract at the end of the day, correct? Hence heads will try to be together because this is favorable interaction. Similar thing can be seen here. All heads are together, all tails are together. Okay. On the other hand, head water interaction, as I have mentioned, it is polar in nature, right? So polar molecules are happy and easily dissolvable in water, indicating that interaction of head group with water is a favorable type of interaction. Hence a membrane, which if lipids are present randomly in water in water, all the head groups will try to come together because of the polar nature, and all the head groups will try to be in water because they have favorable interaction with the water. Okay, the last interaction is the van der Waals interaction. Again, it is the hydrophobic effect, right? So we can say the acyl chains, they are hydrocarbons at the end of the day, right? This is a very easy, simple representation of a hydrocarbon chain which is present in an amphipathic molecule. So these will try to stay together, and hence again they will try to orient themselves in such a way that all the tails are facing towards same side with each other. Because of head interaction, head water interaction, all the heads will try to stay together, leading to the formation of a stable structure where all the heads or the polar entities will face water and will be stabilized, while all the tails or the hydrocarbon chains will stay together and will be stabilized. Okay, now having this knowledge, now this representation is more easily understood, correct? If we look at the basic cartoon representation of a membrane, which we are following since our first lecture, you can see the head groups are closely packed together. Now why they are closely packed together? Because they are polar in nature, right? They love to be in water, and hence this is a favorable orientation. On the other hand, these tails, they are happy in an environment where there is no water, and they are among themselves, and they are stabilized by van der Waals interaction and also hydrophobic effect. Okay, So this representation is energetically favorable, and hence as making such huge structure or a membrane which can withstand so many proteins and it's functionally very dynamic, making this structure stable, all these forces play major role. Okay. As we know, as I have discussed in the last slide, that why there are no covalent interactions present. If the covalent interactions are present for the stabilization of a membrane, it will be a rigid structure and it won't be as flexible as it is required for its functionality. Okay, this can be easily understood if we look at the energies which are associated with the bonds. So even if a covalent bond is present for, say, one can argue that the system can provide the energy for the covalent bond to be broken down, and hence the fluidic nature or fluidic property of the membrane can be retained. But if we look at the energies which are associated with the covalent bond, you can clearly see that a covalent bond is require 400 kilocalorie of energy to break one covalent bond. This is a huge energy for a system, especially for that system which is very dynamic, where there is lot of motion, a lot of changing of place, and very frequent adaptation to the stimulus is required. Okay, if we look at hydrogen bond, it is 12 to 16 kilojoules. Permanent dipole, those polar head group interactions, it is 0.5 to 2. On the other hand, van der Waals interaction energy for van der Waals force or van der Waals type of bond is less than 1 kilocalorie. Hence, majority of the interactions which are present in stabilizing the membrane is coming from van der Waals interaction or hydrophobic interactions. Though their dissociation energy is very low, one again can argue that then how they are stable at the end of the day, right? If this is so little amount of energy is required for them to break, how come they are the ones which are the major contributor for the stabilization of a membrane? Well, the answer lies in the quantity or the number of such interactions present. One covalent bond requires a lot of energy to be dissociated, while if we combine a lot of van der Waals interaction or weak intermolecular interactions, they actually, though individual interaction or single bond energy is very less, but if we increase number of such interactions, the system actually becomes stable. Okay, I hope it is clear to you guys. Okay, so now as we are talking a lot about lipids interaction, and we now know that our membrane is made up of lipid bilayer. It has proteins, it has cholesterol and lipids also, and the major type of lipid which is present in a membrane is called phospholipid. But what exactly are lipids, right? Why they are so important, and why basically they are literally making a cell, or in a broader perspective we can say that they are major contributors for the survival of, or for just being complex organisms. Okay, so if we say a basic definition of a lipid, so lipid is derived from Greek word lipos, meaning fat. Therefore, lipids are by definition insoluble in water but soluble in non-polar organic solvents. Water insolubility is conferred via lipid molecular structure, having large region of the surface composed of hydrocarbons with very few polar groups. I hope all of you remember your chemistry 101, that this is very basic, right? If there are no polar commodities associated with a macromolecule, it will be non-polar molecule. So non-polar molecules in chemistry, as we know, that like dissolves like. If a molecule is non-polar, it will be happy in or dissolve easily in non-polar solvents. While the solvent which we are referring here, as I have said that we are majorly water beings, so water is present and water is polar in nature. Hence, lipids are not soluble in water, this is somewhat of a mystery, right? We are saying the lipids are essential for our survival or just for being a macroorganism. The complexity which is associated with cells and their combination making coming together and giving rise to a organism. On the other hand, we are saying that we are water beings, or our solvent, our universal solvent is water, yet lipids are not soluble in it, correct? So it's a very fascinating area of research in understanding different types of lipids. Oh, I guess I've my slides are not sharing. Yeah, I hope you can see my slides. So this is a very basic introduction of lipids, which I'm gonna share these this particular video with you guys. Let me share again. I guess you guys can't see anything.
अरे यहाँ तो उतार ये काला चश्मा। इसे तो उतार दूं, पर इस टैन को कैसे छुपाऊं? धूप में आता है, फेस वॉश से भी नहीं जाता है। सिर्फ फेस वॉश नहीं, एवरयूथ नेचुरल्स टैन रिमूवल स्क्रब भी कर। ये डेड स्किन सेल्स हटाए, टैन घटाए और स्किन ब्राइट बनाए। एवरयूथ प्योर स्किन हैप्पी हर दिन।
I hope you guys can see and hear it.
Today, we're going to talk about lipids. Lipids are an integral part of every cell membrane in every living organism. Looking closer, you can see that lipids are part of the phospholipid membrane that is the boundary of every single cell. Most people think of lipids as fats, such as the fat in your body. Fats provide long-term energy storage and insulation in living organisms. But fats are just one type of lipid. Oils, waxes, and steroids are also types of lipids. Examples of steroids include cholesterol and hormones such as testosterone, which is produced in the testicles, and estrogen, which is produced in the ovaries. A common feature of all lipids is that they don't dissolve in water. So, what makes something a lipid? All lipids are organic macromolecules. This means lipids are large molecules containing the element carbon. Lipids also contain hydrogen and oxygen. Organic macromolecules, such as lipids, are formed by many units called monomers that are chemically bonded together. In lipids, the typical monomer is something called a fatty acid. A fatty acid contains a chain of carbon atoms attached to each other. Hydrogen atoms are also attached to these carbon atoms. You may recall that carbon can form up to 4 covalent bonds with other atoms. When each carbon atom forms 2 single bonds with adjacent carbon atoms and another 2 single bonds with adjacent hydrogen atoms, we call this fatty acid saturated. This means the fatty acid is saturated with all the hydrogen atoms it can possibly contain. Because of this structure, saturated fatty acids are straight molecules that can pack tightly together. As a result, saturated fats are usually solid at room temperature. Examples of saturated fats include lard and butter. In contrast, unsaturated fats are usually liquid at room temperature. These include things like vegetable oil and olive oil. So, why are unsaturated fats liquid at room temperature? Well, unsaturated fatty acids have one or more double covalent bonds between carbon atoms. This means the unsaturated fatty acid has gaps in the hydrogen saturation. The carbon double bonds form kinks in the fatty acid, which prevent the molecules from packing together tightly. As a result, the more loosely packed molecules form a liquid instead of a solid at room temperature. Here's a tip to help you remember which type of fat is solid or liquid. Use the letter S at the beginning of the word saturated for solid at room temperature. Now that we've talked about fatty acids as lipid monomers, what is a lipid polymer? A lipid polymer, called a triglyceride, is formed when three fatty acids bond to a glycerol molecule. A triglyceride is saturated if it contains only saturated fatty acids. And a triglyceride is unsaturated if it contains any unsaturated fatty acids. To sum up, all living organisms use lipids in all of their cell membranes, as well as for long-term energy storage and insulation, and in hormone formation. Lipids are organic macromolecules containing mostly carbon atoms, as well as hydrogen and oxygen atoms. Lipid monomers are fatty acids. Saturated fatty acids are saturated with hydrogen because their carbon atoms form only single bonds. As a result, saturated fats are solid at room temperature. In contrast, unsaturated fatty acids have gaps in their hydrogen saturation because their carbon atoms form one or more double bonds. As a result, unsaturated fats are liquid at room temperature. Lipid polymers are called triglycerides. Saturated triglycerides contain only saturated fatty acids. Triglycerides are called unsaturated if they contain any unsaturated fatty acids.
Right, so I hope that video was pretty easy to follow. It was just a basic introduction of lipid, their structure, especially related to the biological membrane, that is the phospholipid. Okay, but why we are so much concerned about lipids at the end of the day, right? So this can be easily understood if we look at the functions which lipids are performing, and I'll tell you one interesting fact by the end of this slide. So basically, lipids, we talk majorly about proteins, right? The moment we say some disease is there or anything which is wrong with our system, we directly go to, especially researchers, we directly go to either protein, our first instinct is protein, or we directly go to the genes, right? Because genes are the one which are having the ability to alter our function. At the end of the day, who are performing all the function? Proteins are the one which are performing all of the major functions. Yet there's a lot of diversity associated with lipids. Like even till now, we don't know how many different types of lipids are present in our system. In living system, till now, we know that there are more than 1,000 different types of lipids present, right? Even in proteins, if you know that we can actually classify proteins based upon their structure, right? But for lipids, it's such a difficult task. Imagine you have more than 1,000 entities having different characteristics. How you can classify them, right? And these are the ones which are identified because they were easily seen, and like all sort of stories are associated with them. What if we are not looking for such major components. So even researchers know that there are, apart from 1,000 different types of lipids which we are aware of, there are other lipids, like more than 3,000 different small kind of lipids which are present in our system. Hence their classification, their role, their function is very oblivious. So till now, whatever we know, these are the major functions which are associated with lipids. Again, you need to understand that this might not be the complete list of functions which lipids perform, because we don't even know how many total different types of lipids are present in the system in the first place. Okay, so lipids the functions of lipids include energy source. We know that fats and oil, during extreme condition, they can act as substrate for energy, correct? Then membrane bilayer component, defining the cell permeability barrier. We are discussing this since the beginning of this course. Matrix for assembly and function of many catalytic processes. This is associated again with the lipid raft model which we have discussed in the beginning of this course. Then chaperones for protein folding. Light absorber like chlorophyll and retinal pigment, they are actually lipidic in nature. Energy transduction, like retinal component attached to the rhodopsin, so it helps in photon energy transfer, and hence we are able to see, right? Electron carrier in the ETS chain, if you remember, there is a coenzyme Q small molecule associated. It is lipidic in nature. Okay, hormones like testosterone and progesterones, they are lipidic in nature. Vitamins such as A, D, E, K, they are lipidic in nature and hence are lipid soluble only. Antioxidants such as vitamin E. Signaling molecules such as ceramide. So if we can see, lipids are actually performing functions in all the area of a cell, right? They are energy source, they are present in the membrane, they are present in energy production mechanisms, they are antioxidant in nature, they are vitamins, they are even signaling molecules, right? So lipid or research in lipids is very diverse, and even their functions are not clearly known till now, but these are the major functions which we know of. As I've mentioned, that if a macromolecule is so complex, their classification at the end of the day is very difficult, right? Yet we can have certain assumptions in place and try to classify the lipids. Okay, so there are majorly different types of classification system present, yet even with those assumptions, most of the lipids, or say all of the lipids, won't be able to be characterized or classified. But for the beginning purposes, if we can see the first table, this one, it basically classifies lipids into 4 different groups, right? So the first group is called non-hydrolyzable lipids, which means they won't be further hydrolyzed. And the type of lipids which are non-hydrolyzable, they are hydrocarbons. They can be simple alkanes or terpenes. Then substituted hydrocarbons like long chain alcohols, long chain fatty acids, detergents, steroids, vitamins. Simple esters like acyl glycerol, cholesterol esters and waxes. Complex lipids such as glycerophospholipids, sphingolipids. Glycolipids such as glycoglycerolipids, glycosphingolipids. In glycosphingolipids, major lipids comes as cerebrosides, gangliosides, and then lipopolysaccharides, right? If we look at major categories of lipids, we can categorize them into around 8 major categories. Fa means fatty acid, as you can see here. Gl means glycerolipids. Gp means glycerophospholipid. Sp means sphingolipid. St means sterol lipids. Pr means prenol lipids. Sl means saccharolipid, and Pk means polyketide. we will see these in at structural level also, because these are the categories or the type of lipids which are present in the membrane. Okay, I guess this is bit too much information with respect to lipids today. We will continue about the classification of lipids and certain nomenclature of lipids, specifically with respect to the lipids which are present in the membrane, in the next class. Okay, till then I'll prefer that if you guys can go through the study material and read something about the lipids, because why? Membranes have two major components, the lipid and the proteins. So right now we have entered into the world of lipids. So in lipids we're gonna study about different types of lipids, the properties which lipids impart to the membrane, and what if those lipids are not present, so what can be the like side effects of such kind of incident if lipids are not present in the membrane. Okay, so we're gonna end our class here today. If you have any query or any question, feel free to ask me on the mail, or you can ask in the chat itself. I'll be happy to answer your questions, and if you want me to change my way of teaching also, or if you want to incorporate some things, I'm open for the feedback, and I'll be happy to incorporate if I'm able to. Okay, so thank you so much for your attention and hope you are enjoying this course. Bye.